All right, hello everyone. I'm Vincent. Um, I guess I can just say a few things about what I'm talking about today. So the first thing is that I will be sharing my screen a lot, and I probably won't see messages that are sent through Google Meet. So like, I would appreciate if you could just say them out loud, or like if one of the mods here could just say any messages
Yeah,
that people comment.
that's what happened last time. If
Oh,
this,
okay.
I'll tell them, or I'll tell you what their question is.
Yeah. I mean, other than that, we can just wait.
Sure, let's wait until we hit like 45 or something. I don't know, settle for an
It's
arbitrary
not
number.
gonna happen, dude.
Yes, it will. It's a strictly increasing function. Maybe a time cutoff is better. Let's say 6:34. Yeah, I just saw it decrease, so I guess it's not strictly increasing. That's unfortunate.
By the way, you guys who are like Everraise or participating in Everraise, can you like give an indication of what subject you're doing? I'm just curious.
Everybody who's an Everest student, can you please send your subject in the chat? Also, we hit 45. I was right. Biology, physics, math comprehension 2, math 1,
Oh,
physics, math 2, math 2, math 2.
yeah, I can see it for now, but like, yeah.
We didn't let the bio kids know that this was happening, right?
Oh,
Yeah, I posted on the forum, so should be
okay. I suppose not all of them are here yet, but it's 45, 46 people, and
yeah.
all right. I think we should start now. Vincent, if you could please introduce yourself and your topic, and then you can go ahead at your own pace.
Yeah. Hello, everyone. I'm Vincent. I'm a first-year student, or I just finished my first year at MIT studying math and biology. In high school, I did a bunch of math contests and participated in the IMO. And I guess my goal for this talk is just like introducing you guys to maybe some interdisciplinary work that you haven't heard of before, because I think that a lot of these applications are really cool. And I just want to encourage you guys to study fields other than just the main one that you're focusing on, because knowledge of other fields can be like super rewarding. All right, so I'm going to share my screen now. Let's see, does this work?
Yep.
Okay, cool. Right. Okay, yeah, I'm kind of like writing text with a mouse, so it's not going to be that great. But anyway, so I guess, right, so this topic is math models in bio, and there's two main subjects we're going to focus on today. The first one is about inbreeding, and specifically, why is inbreeding bad? And I'll clarify what I mean by this later in case you guys aren't exactly sure what I'm talking about. And the second one is how do transcription how do transcription factors locate DNA sites? So yeah, if you haven't heard of like what all of these terms mean, don't worry, I'm going to introduce them later. All right, I guess we can get started by talking about inbreeding. So the most famous example is like in the 16th and 17th centuries, there was this royal family in Europe called the Habsburgs. And the way they consolidated power was like, their goal was basically get a female who owns a bunch of land and a male who owns a bunch of land, make them have kids together, and that way like one of the kids will inherit the land from both parents. And the goal was like this way one of their kids will eventually have like a really large amount of land and be a really powerful monarch, right? So this sounds like a good idea, except what ended up happening after a few generations was like they noticed that their kids started having these really deformed jaws and a bunch of other traits, and this became known as the Habsburg jaw. So the question is like, why did this happen? And like in general, are there like bad parts of inbreeding that we should like avoid? So you might be thinking this is like sort of an obvious question, like of course you shouldn't marry your relatives, but I would argue it's like not that intuitive, because if you imagine that like you marry your relatives, so you guys share a bunch of DNA, right? And what that means is that you have a lot of genes in common, which means like since there's less genetic variation, basically you have like less exposure to bad genes, right? So it's not obvious at first that inbreeding will lead to a bunch of bad traits showing up. All right, so to study exactly how this works, I'm going to introduce pedigrees, which you probably have seen before. So basically the way pedigrees work is it's just a model of family trees. And so these circles represent females, these squares represent males. So this horizontal line means that these two people have kids together, and this vertical line means that this is like the kid they have. So those two had a kid, these two people had a kid, and let's say they have a kid together. So this is like what normally happens when you have a 3-generation family, right? Like this is a typical granddaughter, and these are like typical grandparents. All right, yeah, so this is what normal pedigrees look like. And we're also going to introduce something called the inbreeding coefficient. And what this is basically probability that a given child's alleles both come from the same parent allele. So to clarify what this means, basically, all right, so we're going to make some assumptions. The first one is that all traits are Mendelian, and that means that there's only like, so Mendelian traits only have like 2 alleles, right? There's the dominant one and the recessive one. And what this means is that if you have a big A, sorry, big A is dominant, little a is recessive. So big A, and big A, little a will produce the dominant phenotype, and little a will produce the recessive phenotype. All right, so what this inbreeding coefficient is like, let's imagine that these parents, so these 4 grandparents have like 8 alleles for a specific gene, right? And let's assume they just all have different letters for now. So A, B, C, D, E, F, G, and H. So we're going to look at the alleles of this granddaughter, right? And so for both, what we mean by both child's alleles coming from the same parent allele is that like both of these alleles that this grandchild has come from the same one of A, B, C, D, E, F, or G, or H. So like we have A, or C, C, or like F, F, I don't know. And obviously this can't happen, right? Because one of this grandchild's alleles will come from this side of the family, and one of them will come from this side of the family. So you're never going to get a situation where this happens. So that's why the inbreeding coefficient of a pedigree where there's no inbreeding is 0. And so this makes sense, right? Like the inbreeding coefficient will only be positive when we have a pedigree with inbreeding. All right, so now let's look at what happens when we actually do have inbreeding. Okay, so yeah, this coefficient is 0. So when we do have inbreeding, I guess there's 2 main examples we can look at. The first one is the simplest one, which is when you have siblings that have kids together, which, by the way, is a terrible idea, but it happens sometimes, so we're just studying it anyway. So, right, let's see what happens in this case. So we're again going to assume that the parent generation, sorry, by parent, I mean the oldest generation. So parent generation here is actually grandparents. All right. So here we have the parent generation, which again has 4 alleles, 2 from each person. And like, what's the probability that this granddaughter's alleles both came from the same parent allele, right? So like, what's the probability that we have 2 A's or 2 C's?
Vincent,
Yeah.
someone's asking, isn't inbreeding in a pedigree 2 lines?
So I don't actually remember. Wait, what do you mean by 2 lines?
A double line, he clarified.
Like this?
I suppose so? Yeah, no, he's taking it.
Okay. Yeah, I think I've seen that in a few places, but not all of them. So probably. You're probably right, but yeah. Okay. So yeah, what happens here? So like, obviously for both of these big A's to result in this child, like this grandparent needs to pass both of these big A's over, right? And then like both of these big A's have to pass down to this grandchild. So the probability of this is like 1/2 to the 4th power, which is 1/16. So this is the F factor of this case where you have direct siblings that are having kids together. Right, so this is actually not as common. Luckily, it would be kind of bad if it was. A far more common example is when you have first cousins that mate. And this actually happens like 1 in every 1,000 cases in the US. What I mean by this is like, if you just take any child at random, there's like a 1 in 1,000 chance that their parents are first cousins. So this is like pretty common and it's not very good. And I'll explain why later, but yeah. All right, so, right, I'm just drawing the pedigree for first cousins right now. And it looks something like this. Now there are 4 generations, and right, so these 2 are first cousins, and this is the child they produced. So like, again, we're looking at the probability that both of this child's alleles came from the same parent. And the only way this can happen is like one of these shared parents passes down an allele in both directions, right? And both of these directions end up in the kid. So actually I should probably use a different color. Right. So what's the probability that this happens? The probability of big A, big A. I mean, it's basically the same calculation, right? Like you need this big A to pass down 1 generation, 2 generations, 3 generations, and you need the same thing to happen on this side. So like there's 6 events where big A has to get passed down. And that means that there's a 1/2 to the 6th chance, or 1/64 that this happens. So yeah, this is the inbreeding coefficient of this pedigree. All right. So why is this relevant for what we're doing? So I'm going to make one more assumption, which is like, assume one of your parents is the carrier for deformed jaws. So what I mean by this is like, we're studying this deformed jaw gene, right? And so we assume that this is Mendelian, and like from the fact that not everyone has a deformed jaw, or like from the fact that this deformed jaw like sort of came out of nowhere, we can deduce that the deformity is recessive, right? Because if it was dominant, then like, I mean, it would just be a lot more obvious where this deformed jaw was coming from. So this is jaw, and this is bad. And so this big A is the normal allele for like having a regular jaw. So I'm going to say this happens like, if you take a random person and look at their alleles, there's like a 1 in 10,000 chance that they have this little a allele for this deformed jaw. And there's like a 1 minus 10 to the -4 chance that they have the normal allele. And this is like sort of reasonable, right? Like when you look in the street, you don't see a bunch of people with deformed jaws. So it's like roughly at this frequency. So yeah, what this means is that for someone to have deformed jaw and have it be visible, they need to have like little a, right? Which is pretty uncommon. So in particular, in this case, where you have a normal pedigree with no inbreeding, the probability of little a is like, it's just going to be 10 to the negative 4 squared, right? Which is 10 to the negative 8. And the reason is that, I mean, we can basically just assume that these two little a's have to occur independently. And yeah, so this is a very uncommon thing. So the assumption we are going to make is that assume one of these parents is a carrier, which means I'm just going to assume for the sake of argument that this grandparent actually has a little a, and like maybe they have big A little a, because almost everyone is going to be a carrier instead of a little a little a. All right. So if we condition on this grandparent having this hidden allele for a deformed jaw, so like what's the probability that this grandchild ends up with a deformed jaw, right? So like, first of all, this little a has to get passed down, but also there needs to be a second little a from this side of the family, right? And this is very uncommon. We know that it happens like roughly 10 to the negative 4. Or like technically there's 4 alleles, so it's like 4 times as likely, but it's on the order of 10 to the negative 4. So what this means is that the probability of little a under this assumption is going to be dominated by this 10 to the negative 4 factor. So like, even if we already know that one of these grandparents has this allele, the probability that this grandchild ends up with a deformed jaw is still going to be really low. So meanwhile, what happens in this case, right? Like if we assume that this grandparent has this little a, then it's way easier for this grandchild to have 2 little a alleles, right? In fact, the probability that this happens is just the F factor, because we're just looking at the probability that both of these little a's get passed down. So the probability is like roughly 1/16. There's also a small chance that this parent also has a little a, but we can disregard that because it's so unlikely, right? So what this means is that the probability of a little a here is dominated by 1/16, which is way more common, right? And similarly here, it'll be dominated by this 1/64 factor. All right, so this is a partial explanation of why inbreeding is bad, but it's not a complete explanation, right? Because this assumption that one of our grandparents or parents has this deformed jaw allele is still happens very rarely. So what I mean is, like, even if we assumed that everyone in the world started having first cousin matings or like sibling matings, the frequency at which you'll see these little a's with deformed jaws is still going to be very low, because it's very unlikely that this happens in the first place, right? Like, there's still like a 10 to the negative 4 chance that any one of your parents has this deformed jaw in the first place. So how do we resolve this? Or like, the question is, why is inbreeding bad in general? Like, even if my family doesn't secretly have this deformed jaw allele, why is it a bad idea to have inbreeding anyway? And the reason is that, well, actually, it's pretty simple. Basically, humans have a lot of alleles / traits / right. So like, humans just have a lot of genes. And what that means is, like, we have like around 50,000 genes in our genome. And most of these are going to be like relatively harmless ones, right? But there's still a decent number of these where this recessive allele will be super deadly and super rare. So even if the probability that any given allele only happens like 10 to the with probability 10 to the negative 4, when you look at all 50,000 of these traits, what you'll end up finding is that the average human has like 7 to 8 of these super bad recessive alleles. So this is the real reason that inbreeding is bad. It has nothing to do with the jaw, because the jaw defect will still happen very rarely. The fact is that even if you don't have this jaw defect, there's still like 7 to 8 other traits that you do have these really bad genes for. And the more inbreeding you have, the more those traits are going to manifest in your kids and your grandkids. So yeah, this is basically why you shouldn't do the things that are drawn on this page. So we can actually take this one step further. So I'm going to ask, like, what frequency of genetic disease cases result from first cousin matings? So we're going to make some more assumptions. I mean, the first assumption is that the only matings are normal matings between unrelated people and first cousin matings. And like I said earlier, this first cousin mating will happen with probability like 1 in 1,000. And this normal mating will happen with probability 1 minus 1 in 1,000. So the second thing we're going to assume is, I mean, it's mostly what we've been doing so far. So there's these super deadly genetic diseases, which are recessive, and the dominant allele happens with probability 1 minus 10 to the negative 4. The recessive one occurs with probability 10 to the negative 4. All right. So if we look at like how these genetic diseases result from first cousin matings, like what's the probability that if you take any child, they have the disease and their parents were first cousins? I mean, first of all, the probability that their parents are first cousins is like 10 to the negative 3 by assumption, right? And even if their parents are first cousins, we need to look at the probability that this little a allele exists in one of their parents. So you can see there's like 4 spots for alleles from the grandparents or great-grandparents here, and each one occurs with probability 10 to the negative 4, right? So we can just assume there's like a roughly 4 times 10 to the negative 4 chance that this little a allele already exists inside the pedigree. And then given that this little a allele exists like up here, the probability that it's passed down to this child is the F factor, which is 1/64, right? So there's a 1/64 chance, given this allele exists and given that we have this first cousin mating, that this first cousin's child will actually have this condition. And this amounts to like 10 to the negative 7 divided by 16. So this is the probability that you end up getting this disease because of first cousin matings. And the probability that you get this disease as a result of a normal mating is like, the probability that a normal mating happens is 1 minus 1 1,000, right? And then when that happens, we saw earlier that the chance that you have 2 little a's is just 10 to the negative 8. So we're just going to assume this equals 1 because the difference is negligible. So yeah, this is just 10 to the negative 8. So these are the cases that originate from first cousin matings, and these are the cases that originate from normal matings. And what that means is like the total fraction of first cousin matings that are producing diseases is like 10 to the negative 7 divided by 16 over the combined total. The combined total is 10 to the negative 7 over 16 plus 10 to the negative 8. So this is the fraction of the diseases the disease cases that are coming from these first cousin matings, right? And if you just evaluate this, it amounts to 38%. So this is like kind of surprising and also super ridiculous, right? Because these first cousin matings are happening so rarely, like 1/1000th of the time, but they're actually responsible for like more than a third of these genetic disease cases. It's just like kind of sad, because what this means is like these first cousin matings are contributing to like so much suffering from these genetic diseases. And if we just avoided having first cousin matings, we could cut down on the number of these diseases by like so much. And for some reason, we're not doing that. But like, right. So this is just something to keep in mind about like where these genetic diseases are often originating from. All right. So I'm going to move on to the second topic today now. And this is just how transcription factors locate DNA sites. So in case you don't know what that is, imagine that this is the nucleus of a cell. So the cell is much bigger, and the nucleus is just the smaller thing I drew. So we have like this long DNA molecule, and like there's going to be some section of this DNA molecule which encodes for this protein X. So when a cell wants to make this protein, basically it's going to send like this enzyme called RNA polymerase. And RNA polymerase is going to like bind to some part of this DNA. It's going to copy this section of the DNA. It'll produce mRNA. And then basically this mRNA gets sent outside the cell. It gets sent to like some ribosome with like tRNA in it. And this ribosome will produce protein X, right? So this is like basically our understanding of how cells produce proteins. So the question is like, what if I want my cell to produce more than a normal amount of protein X? Like maybe there's some condition going on where I need to produce twice as many of these proteins. So in this case, the cell will send a transcription factor, and this transcription factor will bind to like a place on the DNA right before the part that codes for protein X. And what this transcription factor does is it makes it easier for the RNA polymerase to bind to the DNA. So this is like increasing the probability that RNA polymerase will bind to the DNA. And because this probability is increased, it means that this section of the DNA will be analyzed more times, and this cycle will, or this process will happen more, so more of protein X will be produced. There's also transcription factors that have the opposite effect, but the process is the same. All right, so the question we're looking at, which is like a pretty normal one to ask, is like, if I have this transcription factor that's stored somewhere in the cell, how does it actually get to this part of the DNA, right? Like, how does it know where to go, and how does it actually end up doing this efficiently? So there's three main constraints we're going to have on this problem. First one is that DNA is super long and has thousands of binding sites. So this transcription factor is only trying to locate a specific binding site, right? And somehow it has to go through like thousands of these binding sites before it can get there. The second assumption we're going to make is that transcription factors travel 100 to 1,000 times slower in DNA than in cytoplasm. So basically, cells have like this liquid interior called like the cytoplasm, and it's very easy to travel through this liquid, as you might expect, but DNA is like more of a solid. So if you want your transcription factor to just like walk along DNA, it'll be way slower than just like swimming along cytoplasm. The third assumption we're going to make is that the transcription factor has to locate the DNA in a few hours, because like cells have to stay alive and they have to produce these proteins relatively quickly, usually in like a day. So this is like a pretty hard problem because these transcription factors are small and this cell nucleus is pretty big and DNA is super long, right? So how do we actually do it? So the way we're going to model this is like, just for convenience, I'm going to assume the cell's nucleus is like 200 by 200. We're just going to assume that this is the starting point of the transcription factor, and it wants to end up here, right? And let's say this distance is like 70. It's like roughly 1/3 of the width of the nucleus. And we're going to assume that this DNA has like length, I don't know, let's say like 50. I forget what numbers I used, but yeah. So there's going to be like this one site within the DNA that this transcription factor is trying to get to. And right, so there's going to be 3 main strategies that we look at for how this transcription factor ends up locating the DNA. The first one is just going to be like random walk. So basically you can imagine that, again, we have this nucleus, we have the transcription factor, and we have this target site. So this transcription factor is just going to walk in like a random direction until it reaches the target site. And I mean, there's some advantages to this strategy. One is that it's super simple. You don't really have to waste energy, and you don't really have to think about where you're going, right? Which is pretty good because these transcription factors are like relatively simple molecules. They can't tell like what's 5 units in front of them. So this is like, in some sense, the only thing they can do. Right, so we're just gonna like walk along this cell or the nucleus until we find the right site. And as you can imagine, sometimes the transcription factor will get lucky. It'll take like a straight line path, which is super short, and it'll get there super quickly. Sometimes it'll take a super long path, right? Like this picture is an extreme example where it starts at the red dot, travels along like almost the entire grid, and then it finally hits the blue dot. But like this looks extremely bad, but like it actually occurs not that uncommonly. All right. So the other thing we need to know about random walks is that they're like super hard to analyze. So like you might have experience with this if like you saw some random walk question on the Amy, right? And like there's a bunch of casework you have to do to get an answer, because there's like no good way to know like what the expected amount of time is to get from one location to another in a random walk, especially if it's 2D or if it's 2 or more dimensions. In 1D it's easier, but like in 2 dimensions or more, it's just super hard. So that means we're just going to simulate it.
Vincent.
Yeah.
A student is asking, what about flood fill or something like that? Can we find path with that?
Can we find, sorry, what?
What about flood fill or something like that?
Oh.
Can we find path with that?
So like,
what they said. Sorry.
Flood fill is like, basically for flood fill to work, your transcription factor has to have like some memory of where it's already visited and where it hasn't visited already, right? And this is like a very simple molecule. It doesn't have memory. It also doesn't have like sensory abilities really, so it can't like see what's in front of it. So like if it was more intelligent, then yeah, this would work. But unfortunately, these are like very low level molecules that we're looking at.
Someone is also asking, what is flood fill? I'm not sure how relevant this is to your talk.
Right. I mean, flood fill is just like, you just go in order, sort of, along some space you're trying to fill. This is a very simplified version. You should Google it if you want. But basically, you just go greedily and you travel along all the spaces. All right, so I have some code for simulating a random walk. We're just gonna run it. I mean, you can read it if you want, but it's like not super interesting. So we're gonna simulate like 100 attempts. And you can see that as these attempts are happening, I mean, there's like a lot of variation in how long these attempts are taking, like the number of steps, right? Like this one is like 500,000. This one is like 40,000. So you can imagine that this sort of inconsistency is pretty bad for the cell because it doesn't, there's going to be like drastic variation in how long it takes for the transcription factor to locate the site. But honestly, like, I mean, there's not much the cell can do about it. Well, all of these strategies are going to be, all of these strategies will have a lot of variation. So I think we're stuck on like attempt 88, which means it's taking a long time. Well, it either means it's taking a long time or my Wi-Fi is bad. So this is not expected. Oh, okay. Never mind, it was just bad Wi-Fi. Okay, yeah, so like the mean time it takes is like 150,000 steps, and the max time is like 600,000 steps. Which is like, I guess, reasonable. So the second strategy we're gonna look at is like slightly more complicated. So we're gonna have this 2D random walk along the cytoplasm. So we're gonna do like some weird 2D block. It's gonna end up at this DNA site. So, and then as soon as it hits the DNA, it's gonna stick to the DNA and do a 1D random walk along the DNA. So this sort of makes sense, right? Because you know that your target path, your target site is on the DNA. So as soon as you hit the DNA, you might as well just like only focus on the DNA. And then you'll do like a 1D walk with a bunch of left-right movements. So, I mean, in theory, this sounds pretty good. The issue is this assumption we made that transcription factors will travel way slower along DNA than in cytoplasm, which makes sense. It's like walking through sand instead of walking through water or something. So like the number of steps that this takes will be much less because as soon as you hit the DNA, you just start staying along the DNA. But some of these steps will take way longer. So yeah, you can see, so like in my code, I just made it so that each of these steps take like 200 times as long. So, oh, oops. So if we run this second approach, you can see like, I guess the numbers don't look that different at first. I mean, we'll like wait for the aggregated statistics at the end. And honestly, it might have been a better idea to run this locally, but whatever. What? Okay, we are having some weird issues with this site. They're just not printing things. Okay. Oh, okay, it finished printing. Okay, yeah. So you can see that this time it took like 130,000 steps on average. Or this isn't the number of steps anymore, it's the times with the extra weight so that walking along DNA is way slower. And the max time is like relatively similar. So this is not a big improvement. And, oh, sorry. This brings us to our third strategy, which is, it's like a combination of 1 and 2. So what I mean by this is, so this is actually like a really smart idea, by the way. Like, I don't know how these little things actually do it, but anyway. So first, we're gonna do a random 2D walk, and we're gonna end up along the DNA, right? This is the same as the previous strategy. And now, instead of like strictly sticking to the DNA, we're gonna have like a weak attachment to the DNA. And what that means is that like, once you're on the DNA, there's gonna be some probability P that you stay along the DNA and you do a 1D walk. And there's going to be some other probability 1 minus p that you detach from the DNA. And then you continue doing, once you detach from the DNA, you continue doing your 2D walk, right? So what this looks like is like, first, the transcription factor is going to find the DNA. It's going to walk along it for a bit. Then it's going to detach randomly. It's going to do some other 2D walk. And then it's going to like reattach. It'll keep doing this, right? Until it like finds the right site. And the reason this is super smart is like, it allows you to basically stay near the DNA without actually sticking to it. And what that means is that you can avoid the extra like factor of 100 or 1,000 that comes from strictly sticking to the DNA. So I'm going to run the code for this third simulation. And there's something I should note, which is that basically the dimensions we use to simulate this are like 200 by 200, right? And this distance was like 70. But in real life, these distances are way larger compared to the size of this transcription factor. And the DNA is going to be way longer. So the results from this simulation are like not going to be that different from each other. But in real life, the difference in the runtimes of these strategies is going to be way more significant. The reason I couldn't simulate it with like 10,000 by 10,000, the reason I couldn't do that was like those simulations would take forever and you wouldn't be able to see them run in real time. Okay, right. So this time the mean was like 100,000 and the max is like still roughly the same. And I guess that's to be expected. So anyway, the idea here is that basically like through a bunch of natural selection, evolution, whatever, over time, these transcription factors have evolved so that they have this weak attachment to DNA. And this allows them to like do a hybrid strategy, which allows them to find the target site of the DNA like way faster. In real life, this is like significantly faster by a factor of like 10 usually. So on a related note, this is like pretty similar to something in like theoretical computer science called randomized local search algorithms. And you can Google these if you want. Basically, we have really hard CS problems like traveling salesman problem, which you might have heard of, and they take forever to solve, right? So instead we do these randomized approaches where there's where like first you try to find a locally optimal solution, and then like with some probability you jump to a less optimal solution and you start over. And we found that for like really hard CS problems, this sort of mixed strategy also ends up finding really efficient solutions. All right, so this is like basically all I had prepared today. I can talk some more about like different applications of math and bio if anyone wants, or I can just answer any questions people have.
Someone's asking, what did the output number? Okay, actually you have it up.
Oh, this is just the number of steps that it took on some attempt of the simulation. All right, so yeah, you guys can just ask whatever questions you want, honestly. Is there a more optimal solution? Well, so the thing about how these sorts of solutions came up is like, we literally had like hundreds of thousands of years of evolution, right? And those hundreds of thousands of years of evolution produced this solution that we're looking at right now through like a ton of trial and error. So what that means is like any solution that you see in nature is going to be relatively efficient because just by virtue of the fact that we're observing it right now, right? So there probably are more optimal solutions, but they're not feasible for like, they're not feasible to take place in like a normal cell. like we could probably come up with a more optimal solution with computers, but that doesn't mean a cell can actually simulate that same process. Can we use randomized local search for protein docking? I don't actually know if this is the case or not, but I wouldn't be surprised if like protein docking had similar mechanisms. As of now, I definitely do more math than bio. So like you might know that I did a bunch of contest math in high school, and I only started getting into bio in like this past year. I did do USICO as well. I did not ever make USICO camp, but like I just did it mostly for fun. Where is this message? Oh, I mean, I don't really care what Joe Mama thinks, so it's fine. What would you want to tell yourself as a high school freshman? Well, this is a rather hard question. I think, I mean, there are several things I would tell myself. The first one is like, obviously just to be more open to trying things other than the things that I've already been trying. So like in my freshman year of high school, there was like this pretty long phase where basically every day in class I would just be doing a ton of math, and like I wasn't paying attention to class or like talking to my classmates or learning anything else. And I mean, I would probably change that if I had the chance. Do you regret spending so much time on competition math? This is also a complicated question because it's like, I wish that I spent some time doing other things. Like, for example, I wish I learned how to make a website earlier or something. But like, even if I stopped doing math in high school, there's no guarantee I would have actually spent that time doing something productive, like building websites, right? Like, whenever you look in hindsight, you're going to realize that there are better ways you could have spent time, but that doesn't mean that if you chose differently at that time, you would have actually ended up doing the things you thought were better. Do you have any tips for conducting research with math and bio? Yeah. So, I mean, I guess the first tip is just going to be like, you need to have solid fundamentals and like, I guess like a lot of discrete math like probability and statistics, and also some more calculus things like differential equations, because a lot of these, basically all of these models that you look at are going to use like probability and calculus, right? So like these are definitely things that you basically need to know to be able to make models. And apart from that, once you have the backgrounds, honestly, you can just like talk to professors at whatever college you end up attending. And the intersection of math and bio is pretty big, so you'll probably be able to find people that want to work with you. What is your AOPS account? It is Tasty Math 75025. So once this loads, yeah. So this is my profile. If you want to like stalk me or something, it's fine. Did you do? Oh, sorry. Do you feel like you apply math more to bio, or you take lessons from bio to math? This is pretty interesting. So like, there's definitely a lot of lessons from bio that I'm inspired by. Like, there's a lot of cool design principles that came up, like the ones we were talking about today. There's also just the fact that like evolution is super cool and the outputs of evolution are like super efficient. So these like sorts of designs, or like how chlorophyll traps light, for example, they have been super optimized over time, and they're just like very well designed in general. So like I definitely take inspiration from those, but honestly, just like the fact that I've been doing math for so long means that I apply more math to bio. Did you do science competitions in high school? I basically only did one science competition, which was Regeneron STS in my senior year. I was a finalist for that, and I enjoyed that a lot. But I didn't do like ISEF or like other science fairs. Is Snorlax your favorite Pokemon? For now at least, yeah, it's been my favorite for a while. I guess my second favorite Pokemon would be maybe Dragonite, because it's like cute and super strong and whatever. Were there any experiences that made you particularly interested in bio stuff? Oh, oops. Yeah. I guess the main thing is that, yeah, right. So the main reason I was interested in bio initially was just that I found it to be super inspiring. Like I mentioned earlier, I did Regeneron Science Talent Search, right? And I ended up being invited as a finalist. And while I was there, like Regeneron had a bunch of talks about how their medicine actually works, how they design those medicines, and like why these medicines are different from other medicines that already exist. And just like the thought that went into those medicines and like all the designing and all the just the entire process of that and like how it changes people's lives, it was just very inspiring to me. And that's actually why I started studying bio in this past year. Why did you decide to study math with bio instead of some other subjects? Yeah, basically the same thing. Would you recommend against doing math research without much background in calculus? I mean, so there's a lot of math research that doesn't strictly require calculus, like, for example, a lot of graph theory or like combinatorics or number theory. But honestly, like calculus is just so fundamental that you should probably learn it first anyway. Like, yeah, it's just important. And you can honestly do both at the same time if you want, but you really should learn calculus. If A is the recessive allele, A is the dominant allele, wouldn't a parent need to be little a? Right. So for the parent to have the deformed jaw phenotype, they would need to be little a, but they can be a carrier if they're big A, little a, in which case they can still pass the little a onto one of their kids. Yeah, some part of this you can do with Punnett squares, but Punnett squares are only for like the most simple use cases usually. What is your favorite piece on cello? This is like a pretty typical answer, but it has to be Dvorak Cello Concerto. Like, it's just objectively good. I don't know. What was your favorite and least favorite class you took at MIT this year? My favorite class was systems biology, which was a bunch of these models that systems biology MIT. Covered a lot of topics similar to the ones that we discussed today. It's also taught by this super cool dude who's knowledgeable in math, bio, and physics. So yeah, that was cool. My least favorite class from my first year at MIT. It's kind of hard. I think I enjoyed basically all of them. Like, the thing about MIT is like, well, the thing about college in general is if you find a class you don't enjoy that much and you can drop it, you should drop it, because otherwise you're going to spend like thousands of dollars and a ton of hours on a class that you don't actually like. Do you have a favorite YouTube video that we should watch? Yeah. The video is, oh, my Wi-Fi is really slow. I think you probably wanted me to like advertise your piano videos or something, but I mean, this is my favorite video, right? So I have to advertise it. It's called Dropbox Intern Gets Dropped. Okay. What did you do for your STS project? Basically, I thought about refugee crises and how we could model them in advance so that you could predict where refugees would go before they actually went there. And I mean, you can look up more about it if you want. It's like, you just go to the STS page for like 2019 and you'll find it. you accept AOPS friend requests? Probably not, sorry. How do you start math research as a high schooler? So, so there's like two main things you can do, I guess. The first one is like contacting a bunch of local professors. If you do this, a lot of them are going to ignore you. So what you should do is like find people who have backgrounds similar to ones you have experience in. And like if you email them, you should be like, hey, I'm interested in doing research with you. These are some similar things related to the field you're in that I've already done or I've already learned about. Because you basically just want them to know that you already have some experience and you're not just a random high schooler who isn't able to do anything. What I personally did was I applied to the MIT Primes research program, but it's like kind of competitive and they don't have that many spots. So if you're not able to get into a research program, you should just contact local professors. Elgar is better. I mean, like I like Elgar a lot too. Yeah. Elgar 4 is amazing. Agreed. Along with this is the transcription factor walk to determine specific target in DNA, sort of like, huh. I don't actually know that much about electron cloud configurations, but my impression is that there's way more math involved for those. It's like I can't actually answer this question, but they don't seem that similar to me at first. So there's like very little physics that we needed to talk about transcription factors, but I imagine there's way more for this electron cloud stuff. But I never took like hard chem or hard physics, so yeah. Do you have any obscure musical recommendations? I have a lot. Let me think of, let's see, ones off the top of my head. I guess if you're looking for pop music, you should listen to, oh, oops, that was an accident. You should listen to this Bruno Major guy. He's like kind of obscure, but he has really good songs. Yeah. Well, kind of obscure, like less than 1 million subscribers. I don't know. Was the STS project successful because of the math or the practical application? So like the only math I did for that project was like the basic design and foundation. The rest was like coding the actual model. So I would say like the practical application, because like that's definitely the part I spent the most time on. And like the math alone was not very impressive. It was like more about the amount of time I spent like collecting data and like actually modeling things. Haydn, I mean, I don't know, like most classical composers, whatever, like you can't listen to them for that long or something. Okay. I see Breaking Benjamin. I don't actually remember what that's referring to. What's the best decision you made in high school? The best decision I made in high school is like staying in public school for me, because there were some private schools or like STEM schools I was considering, but ultimately I think if I went there, I would have ended up not expanding my focuses as much. I also liked the orchestra at my local high school a lot, and if I went to a private school, I definitely wouldn't have had that. But this is not like advice that's applicable to everyone. So like if your local public school sucks, then yeah, you could still consider transferring. Can expected value be used on the protein transcription random walk? I mean, so like I was saying earlier, random 2D walks are like super hard to analyze. And the thing about cells is like real cells are 3D, they're not 2D, right? The thing I drew here was just a simplification for today. But with 3D, these expected values are even harder to analyze. So like honestly, we mostly just simulate things. We have like some asymptotics for what happens in the long term for 3D walks, but those aren't that helpful here. Basically, like, these random walk questions, they seem super simple, but they're actually like almost impossible to do actual math with. Did you join any youth orchestras in high school? Other than the high school I was in, not really. I'm thinking about doing some math modeling related to coronavirus. Yeah, you should definitely learn calculus, but like, even if you weren't doing this math modeling project, I think you should still learn calculus. Just like, it'll just give you a way better sense of like, just how numbers work. And like, if you have some random function, like how do you actually analyze it or know if it's like big or small, right? Like you sort of just fundamentally need calculus to do these things. Have you ever used Punnett squares, complex situations? No, I have not, sorry. Explain roughly how your script works, how it outputs the number of steps. Right. Oh God. Okay. So basically at each step, we do like some random walk or some random step, and then you increment the step counter by 1. And then at the end, you print what the actual value of the step was. Similarly for the other 2 scripts. You just, yeah, it's not as complicated as it looks. What recommendations do you have for future high schoolers? I mean, this is hard to answer. Like, generic high schoolers, I don't really have any recommendations for. The fact that you're at Everraise probably means that you do some STEM subject and like you're pretty passionate about it. I mean, it's just really hard to give this sort of recommendation without more context. But like, in general, I don't know, just like, just don't do things that you think are like a waste of time or you're not getting anything from. And like, if you like something like bio or math or chem and there are like people are telling you not to do it, unless they have really good reasons, you should just do what you like anyway. St. Mark's Orchestra, blah blah. Do you suggest learning calculus or graph theory first? Probably calculus. It's just more fundamental and like, oh, sorry, you said group theory. Group theory is honestly like impossible. Like, you could spend the rest of your life learning group theory, and like at the end, you still wouldn't. You would have like an okay understanding of it, but like you would probably still think there's more to learn. With calculus, it's like there are very hard calculus things that are research subjects. But what I mean by learning calculus is just like the fundamental techniques. And I mean, you can get that done pretty quickly. I moved to another state this summer. Okay, I'm not sure why you're telling me this, but like, I can look at it later, I guess. I made a program in Java that analyzes amount of test kits saved with block testing. Would this make a good science project? So honestly, it depends on how legit your science fair is. There's actually already papers that have been published that have analyzed the same question about coronavirus and block testing. And like, if we test people in blocks, we can save on the number of tests we do by like a large amount, right? Like, legit science fairs will not care about this probably because it's already been done, unless you have like a very innovative approach. But like, you could try submitting it to science fairs anyway. I honestly don't know. What did you have for breakfast? I did not eat anything actually, because reasons. Yeah. Group theory is impossible, agreed. Yeah, the graphical nature of calculus is like, it definitely helps a lot. You just see graphs and you visualize them, and you like know which things are like large and small. What advice do you have for doing a research project? The main one would be to give yourself a lot of time. Like, if you think you want to finish a project by some date, you probably want to start like a year earlier or something, or like start looking for the projects a year earlier, because it'll take a while to actually find a project, and then it'll take a while to read through papers and things and figure out exactly what you want to research. I think a lot of times in high school, people underestimate how much time they'll have to spend on research by a really large amount. Which is better, EI, pi, 1 or 0? Probably 1. I don't really care about these other fancy numbers, like, other people can go debate them for me. I don't know. What are you planning on doing fall 2020? My God, I don't know. I'll definitely be learning a lot. It might be through school and it might not be, but who knows exactly what. What's your favorite non-integer? 2.5, I don't know, dude. All right, I guess that's it for today. So thank you guys for coming, and also thanks Everraise for having me.
Thank you so much, Vincent. Let's have a round of applause, or as best as we can have one over Google Meet. There's also talking in the chat. Thank you.